Welcome to lesson 3B in our heredity and genetics grid. This is about Mendel's laws. So our friend Gregor Mendel studied these plants for 8 years and came up with a ton of research and presented most of it. But funny enough, not most of his studies were not even taken seriously until maybe 50 years later. And a lot of them were then put together, and a lot of these laws were named after his death. So just think of how we can end up with so many different traits when we might have the same set of parents. Have you ever looked at your siblings or your cousins and family members and wondered how come we have some things in common but not others? I always like to say, look at it like a deck of cards. And if you cut the deck of cards into two piles and then shuffle them together and deal out 5 random cards, sure, there's a chance you'll get 5 spades in a row. But there's also a chance that you would just get one of those spades. And they do it again, shuffle the deck again, and deal it again, and you might get that same spade. But the chances are pretty low. But there's a chance that you might get at least another spade. And it's really every time the DNA is mixed up and every time it's recombined with another set of DNA, it is like shuffling a deck of cards with way more than 52 different options. We'll talk about more of that later on as we go through these notes. But first, I want to talk about some common phrases I'll be using. Some of them you've heard of, and maybe some of them you haven't. The first table on your note-taking guide you're going to fill in is about the difference between genes and alleles. A gene is a piece of the DNA, we've talked about this, that contains a certain set of instructions for a certain inherited trait. And we know that it's part of the chromosome, it's made of DNA, it's sections of the DNA are each different gene. So this might be the gene for eye color, this one might be the gene for height, and so on and so on. There's hundreds of different genes, maybe thousands of different combinations. So there's your definition, and we haven't come to the analogy yet, but that'll be on the next slide. The definition of an allele is it's one of the alternative forms of a gene, and it determines that inherited trait. For example, if this is the gene for eye color, then one allele for this eye color gene is blue eyes, and another allele is brown eyes. In this example, these are 2 chromatids, and we're talking about flower color in peas. So this might be the allele for purple flowers, and this is the allele for white flowers. And of course, you know that if one of those is dominant over the other, then we would only see that dominant trait. And you might remember which one is dominant from our last note-taking guide. So here's another way of putting it, and I'm going to do another video inception, a video within a video. And I like how this video explains an analogy for genes versus alleles, and you can put this in your note-taking guide too. Oops. There we go. So as you saw, if the analogy is that there's a gene called Coca-Cola, the alleles for Coca-Cola are Diet Coke, Cherry Coke, caffeine-free Coke, Coke Zero, all the different varieties of that gene. And we've already talked about the eye color example, but I like that analogy of different types of Coke products. Now, there's two words here you may not have ever heard, genotype and phenotype. The definition of genotype, it is the entire genetic makeup of an organism, or it's the combination of genes that they might have for a specific trait. The entire genetic makeup or a combination of genes for their trait. Phenotype is the organism's physical appearance because of their genotype. And I like to remember it, the PH here for physical and phenotype. That helps me remember which one is which. And of course, genotype starts with gene. So here's the example. We write the genotype, if we're talking about one trait, we write them as letters. And if it's a dominant gene, we capitalize the letter. And if it's a recessive one, we lowercase. And it doesn't matter if it is A, B, yet Y, L, Z. It just, you write a letter, you choose a letter for a certain gene and genotype, and this is how you would abbreviate it. And this would mean that this is yellow peas because it's dominant. Both of them are dominant, so you know that the pea would be yellow is the phenotype. Even if it had the recessive gene for green, it would still be yellow because yellow is dominant. And if the pea plant had both A recessive genes, then it would be green. And the next term you need to know is homozygous versus heterozygous. Homozygous is when the genotype has two matching alleles, because you remember homo means the same, like homologous that we've studied in our previous level. So if it's got two matching alleles, we call it homozygous, and if it has two different alleles, we say heterozygous. Hetero means other or opposite sometimes. For example, if we have these two allele pairs and they're both dominant genes, and it doesn't matter what the gene is, we say it's homozygous. And if it's two recessive genes, we say homozygous. But if it's a mixture of one of each, we say heterozygous. Now, the heterozygous is sometimes called a hybrid trait because it's a mixture of two different traits, one dominant and one recessive. And of course, I have to throw in my favorite pun about heterozygous genes. These are heterozygotes. Get it? So, now, let's get into Mendel's laws. On each of the next few pages, you're going to write the law. There's three of them. You're going to write the name of the law and a brief shortened version of it. I can help you shorten it if you like. And then there'll be a little diagram or a drawing that you're going to fill in and label. The first of Mendel's laws is called the law of segregation. And law of segregation is that each inherited trait is defined by a gene pair. Parental genes are randomly separated, that's what segregation means, to separate, to the sex cells, so that sex cells contain only one of those gene pairs, and then the offspring will therefore inherit one of the alleles from one parent and one from the other. So, to summarize, it's just the fact that all genes have two alleles, and each sex cell or gamete gets one of them. They're separated for the next generation. In this example, it's an imaginary species of little lumpy creatures. The gene for more lumpiness is capital L, and that's dominant. And the gene for less lumpiness is little l, which is the lowercase l, less lumpy. And when you have two heterozygous lumpies with the genotype big L, little l, that means they have one dominant allele and one recessive allele. If they mate and have children, then the results could be this. Now, of course, this is if they have 4 children, but these are the 4 options for these creatures. Of course, it could be luck of the draw. They could get all 4 of them having this genotype, but these are just the options. So we could see that the big L could be distributed to these two, and this big L distributed also to these two, and the little l to these two, and there's a 25% chance that one of them would be both little l's. So you would see that this little guy is less lumpy than his brothers or sisters, even though both parents were very lumpy. So this is an example of that second experiment that Mendel did, where he crossbred two yellow peas and found out that 25% of the offspring were green. In the Wheatley family, we actually have a perfect example of this, that I have brown eyes and Mrs. Wheatley has blue eyes. And 25% of our kids, one of our four kids, has blue eyes. I like to say, but even though she's got the homozygous gene and I have the heterozygous, it just helped me know that I at least had the little b blue-eyed gene. Now, in Mendel's laws, second law, the second law is called the law of independent assortment. This is that genes for different traits are sorted. That's the assortment part. They're sorted separately from one another so that the inheritance of one trait is not dependent on the inheritance of another. And that kind of sounds a little confusing, so I think when I explain it in this example, it'll make more sense. But here's the definition that you should write or shorten and summarize. Here's the example. Let's see how we have um, two organisms here that are both heterozygous. Actually, we're looking at two different traits, so they're both homozygous. These traits are both homozygous. But just from looking at them, you can't always tell what kind of traits their offspring could have. Um, so these two both, one of them has many lumps. That's the double P. This one also has multiple. And then, I'm sorry, I'm looking at the wrong letter, not P, L. Multiple lumps is dominant. Few lumps is recessive. Then there's purple versus pink. So this one has, these are little p's. I think it's hard to tell. And these are big p's. Pink, purple. If the genes for color and head shape did not assort independently, then only combination of traits already seen in the parents, so basically pink and lumpy or purple and less lumpy, would be possible. But we see in this square down here called a Punnett square, we can see that their possible offspring, there's many possible traits that don't even show up in the parents or the next generation even. And the way that works is you sort them out by different options. So if these two offspring have big P, big L, little l, and both of them have that, then there's still many options. You could have big B, big P, big L, big P, big L, little l, and so on, so on, so on, so on. And look, of course, in the end, you still have a 25% chance of having some that are pink, and you have a 25% chance of having some that are less lumpy. They're just not all clumped together like these guys are. So as you can see, this is why they're calling it the law of independent assortment, that all these traits are sorted out over many generations, and there are many options and many possibilities that come from something, even though that it seems like there's really only a few options. The third law is called the law of dominance, and probably the easiest one to understand, fortunately. The law of dominance is that an organism with alternate forms of a gene will express the form that is dominant. So that's pretty obvious. Like, take these two. We might see that these two true reading lumpy people here, if you just look at them, you can't tell which trait is dominant. If we've never studied their genotype, we wouldn't know. The only way to know is to do like Mendel did, is to crossbreed them and see what happens. And we see when these two cross together that we at least get one purple one, if that's just the first attempt, and we could do multiple tests, of course, and see that until we find out that, okay, this purple gene must be dominant. And same thing with the multiple lumpiness is dominant. Now, there are some lawbreakers, as always. These laws don't always work for some traits, and there's some traits that kind of defy our written rules. We call them, the reason this happens is because they're with what's called a linked gene, because they're linked to a certain chromosome, a certain part of the chromosome and a gene that would only show up if this chromosome was present. Here's an example. The easiest example is what's called a sex-linked trait. So if you look here, the father, whatever this trait is, it doesn't have to be, we don't have to specify one right now, but let's say it's a genetic disease. The father doesn't have it. He's unaffected. The mother has it, but it doesn't. It's only she's carrying it. It's recessive, and it's just not showing up in her because she's a female. It's This one would be a sex-linked trait. Now, all these options of different possibilities of offspring, we could have an unaffected son who, just like dad, doesn't have the gene. We could have an unaffected daughter, just like dad, doesn't have the gene, but is a female. Now, one daughter option could be that she is a carrier, just like mom. But if dad and mom both had something to contribute with this gene, I'm sorry, no, if the dad doesn't present this part of the gene, he gives a different part, then we could get an affected son, and he is not just a carrier, he actually has that genetic condition. There's a lot of different genetic disorders. There's Down syndrome. There's I'm drawing a blank. Hold on. My friend. I'll come back to it. Here's another example. Not such a serious one. This one is called color blindness, and it is widely common with boys. And boys are more likely to develop this trait because the only way a girl can develop it is if she has both of the alleles from parents. Boys just need one of the alleles. And if the female is heterozygous for the allele, then neither she nor any of her daughters will get the trait. But if there is a 50% chance from her that her son could be colorblind if the father is unaffected, even if the father is unaffected. So here's an example. If you can see these dots and see a number in the middle, then you are not colorblind. A colorblind person would not see those numbers that are in green in the middle there. Now that's like a red-green type of colorblindness, and there's other different types of colorblindness, but it's a simple test that people have done. Just a quick scan to see, can you see a number in there? And sometimes kids look at it and they start overthinking it. Just kind of let your eyes blur and take a step back if you're not sure. But if you can see it, write the number down in your notes. Okay, that's all we have for this note, and I hope you understood everything. Let me know if you have questions.